On today's episode, we'll be discussing biochemistry. Biochemistry is the unit where we learn about macromolecules, including carbohydrates, proteins, lipids, enzymes, and nucleic acids. Biological reactions are those that are associated with biological processes and occur with biological molecules. We first have neutralization, there's oxidation reduction, and there's condensation and hydrolysis reactions. We'll specifically be looking at condensation and hydrolysis. A condensation reaction is when two molecules bond to form a larger molecule and water. A hydrolysis reaction is where one larger molecule is separated into two molecules using water. So before we get into specific macromolecules, let's look at macromolecules as a whole. So macromolecules are polymers, which are long chain-like substances composed of many smaller molecules linked together by covalent bonds. And those smaller molecules are called monomers, which can exist individually or as units of a polymer. Carbohydrates are a type of biological molecule that always contain carbon, hydrogen, and oxygen. A monomer of a carbohydrate is called a monosaccharide, and these are basically sugars. We have galactose, glucose, and fructose, which are all isomers of each other, and that means that they all have the same formula but different structure. A disaccharide is when two monosaccharides form together to form the disaccharide. Now let's get into polysaccharides, which are multiple monomers formed together through glycosidic linkages. Some examples of polysaccharides include glycogen, starch, and cellulose. So moving on from carbohydrates to lipids, lipids are composed of carbon, hydrogen, oxygen, similarly to carbohydrates. But lipids have fewer oxygen atoms and significantly greater proportion of carbon to hydrogen bonds. Lipids are hydrophobic and do not dissolve in water. Therefore, they do not dissolve in the aqueous environment of the cell. While carbohydrates provide us with direct energy, lipids store energy in hydrocarbon chains. We're going to look at two different types of lipids. First, we have triglycerides, which are lipids used for energy storage. They are composed of one glycerol molecule and three fatty acid molecules. The linkage of triglycerides is called an ester linkage. A monomer of a lipid is called a fatty acid, and a fatty acid is either saturated or unsaturated. A saturated fatty acid has no double bonds between carbons, therefore it is solid at room temperature. An unsaturated fatty acid has one or more double bonds. When it has one double bond, it is called monounsaturated, and when it has two or more double bonds, they are called polyunsaturated. An example of an unsaturated fatty acid would be olive oil, and an example of a saturated fatty acid would be butter. Now let's get into phospholipids, which differently from triglycerides have a phosphate group. Thus, they have two fatty acids, one glycerol, and its phosphate group, and they are held together by ester bonds. In structure, a phospholipid has a head, which is polar or hydrophilic, and it has two lower tails, which are nonpolar or hydrophobic. Because of the composition of phospholipids, they are able to make up the cell membrane. You will hear later in this video how phospholipids make up a lipid bilayer, which is the main component of the cell membrane. With a basic understanding of lipids, we can now go into proteins, which represents an extremely diverse type of macromolecule. Amino acids are monomers of proteins. They contain a central carbon atom that is bonded to a hydrogen atom, an amino group, a carboxyl group, and an R group, which is also called the side chain. A peptide bond is what attaches amino acids together to form a protein. Let's get into the levels of protein organization. First, we have primary protein structure, which is a simple linear sequence of a chain of amino acids. With secondary protein structure, hydrogen bonding of the peptide backbone causes the amino acids to fold into a repeating pattern. With secondary structure, the protein might form a coil-like shape called an alpha helix, or it might create a folded-like shape called a beta-pleated sheet. In the third level of protein structure, the protein forms a three-dimensional structure from a complex process of protein folding. The fourth level of organization involves a protein consisting of more than one amino acid chain. Now let's get into enzymes. Enzymes catalyze biological reactions. The use of an enzyme-substrate complex is what helps to catalyze a chemical reaction. The combined structure of an enzyme with a substrate that is bound to the enzyme's active site allows for the substrate to separate into two smaller molecules. However, in some cases, the enzyme cannot function properly to catalyze biological reactions. This is because of an inhibitor. In competitive inhibition, the competitive inhibitor interferes with the active site of the enzyme so the substrate cannot bind. In non-competitive inhibition, the allosteric inhibitor changes the shape of the enzyme so it cannot bind to the substrate. So now that we have learned about carbohydrates, lipids, proteins, and enzymes, we now have these nucleic acids, which are also macromolecules. However, I talk a lot about those in my genetics video, so if you want to learn about those, please go check that out. In this unit, we also learned about the cell and its organelles. However, we learned about organelles a lot in previous years, so I'll be skipping that part and going straight to the cell membrane and transport. The cell membrane separates living cells from its non-living surroundings. It is selectively permeable, meaning it allows some substances to cross more easily than others. Hydrophobic and hydrophilic things enter differently. This is because the cell membrane, as previously stated, is made up of phospholipids. Phospholipids form a lipid bilayer by attaching to each other and concealing the hydrophobic tails from the aqueous environment by essentially creating a sandwich. The hydrophilic head is facing outwards, while all the hydrophobic tails are in the interior of what becomes the lipid bilayer. The hydrophilic and hydrophobic layers restrict the passage of many substances, and this is important to the selectively permeable cell membrane. Individual phospholipids can move in the bilayer, which allows for cell flexibility. The fat composition of the cell membrane can vary. The more saturated fatty acids in the membrane make it more viscous, where if there's more unsaturated fatty acids, it can be more fluid. The cell membrane does not only consist of the phospholipid bilayer, but there are also proteins and other molecules embedded into the bilayer. There are a few different types of membrane proteins. First, we have peripheral proteins, which are loosely bound to the surface of the membrane and act as cell surface identity markers. The two other types of proteins we'll be looking at are integral proteins and transmembrane proteins. Integral proteins penetrate the lipid bilayer, usually across the whole membrane, and transmembrane proteins are transport proteins, which act as channels or pumps. Proteins can serve many functions in the cell membrane. It could act as a transporter, it could be used for enzyme activity, act as a cell surface receptor or a cell surface identity marker. It can be used for cell adhesion, or it could attach the cell membrane to the cytoskeleton. Although carbohydrates cannot be embedded in the cell membrane, they play a key role in cell-to-cell recognition. Now that we know all the components of the cell membrane, let's talk about diffusion. Diffusion is the movement of molecules from high to low concentration. So materials can't just move freely in and out of the cell through the cell membrane. This is why the cell membrane is called selectively permeable. There are protein channels that allow specific materials across the cell membrane. So passive transport, also known as simple diffusion, involves the diffusion of nonpolar hydrophobic molecules or lipids, and they can just move through the cell membrane easily without a pump. Facilitative transport is the diffusion of polar hydrophilic molecules, which have to move through a protein channel. Active transport, on the other hand, is quite different because it requires energy. It moves molecules from low to high concentration, so it goes against the concentration gradient. In order for larger molecules to make it into the cell, endocytosis occurs. There are three types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. So we talked about diffusion, but there's also osmosis, which is the diffusion of water from high to low concentration. The direction of osmosis is determined by comparing the total solute concentrations. When a cell is hypertonic, it contains less water and more solute. When a cell is hypotonic, it contains less solute and more water. And when it's isotonic, it contains equal amounts of water to solute. A plant cell is in its healthiest state when it's hypotonic, meaning it has more water than solute, while an animal cell is in its healthiest state when it's isotonic, meaning it has equal amounts of water to solute. That's all for today's episode on biochemistry. We learned about condensation and hydrolysis reactions and all the different types of macromolecules, including carbohydrates, lipids, proteins, and enzymes. And we also learned about the cell membrane and its transport system. Stay tuned for our next episode on cellular respiration.